Is what?
I'm still on chapter 10.
Oh, it's still not from last week.
All right, so we talked about the cell cycle. Interphase consists of 3 stages. What are they?
Interface?
Yep, interphase.
Oh, God.
S1.
S1, S2,
G1,
G1.
G2, and S.
Wow.
Or no, it's G1.
G1,
S.
S.
G1S and
Yeah.
G2.
I watched the video.
Yeah. Okay, good. And then after G2, what's the next stage?
Oh, prophase.
Metaphase. Oh,
PMAT, prophase.
yeah, prophase.
Metaphase,
anaphase,
telophase.
Yep, and we call that mitosis.
And then there's the C one.
And there's the C1. What's the C1 called?
Cytokinesis.
Very good. All right. So we're going to talk about how that cell cycle is regulated. Still roughly incomplete, but we know that there are irreversible points. Like after DNA has been replicated, the cell is committed to cell division. And then also during the separation of sister chromatids, where it becomes, it starts off as an X and then they pull away into 2 sister chromatids. So again, that's another irreversible point. It's committed to finish cytokinesis. So we'll talk about some of these checkpoints in this section. So brief history of cell cycle control. One of the first, I don't want to say hormone, but chemicals responsible for regulating the cell cycle is MPF. And how they stumbled across it was looking at frog oocytes, which is basically like frog eggs. And what they did is they arrested the oocytes at the G2 stage. So after G1 where it grows, and then S stage where it replicates, and G2 final preparations for it, they halted it, and they noticed that there was this really high concentration of MPF. It varied throughout the cell cycle, low in early G2, and then right after G2 was completed, it peaked during mitosis. And they realized that MPF is kind of responsible for this cascading effect to go through mitosis. So here are the 2 scientists that stumbled across MPF, and it's called the mitosis promoting factor. So that's what MPF stands for. And then what they realized was that if they were to take away the MPF inside an egg cell, the cell would not divide. And if they were to insert it into an egg cell, then it would divide. So there's also another chemical involved with mitosis. So it's not only MPF, but cyclins. And so research or discovery of cyclins, they noticed that it would peak again during mitosis, and it went along with the cell cycle. So here you can see that it peaks with mitosis, and then as soon as mitosis is complete, it drops off the mat, or the map, I meant to say map, and then increases again in mitosis. And so there are 2 forms of cyclins that cycle very differently. I know I haven't discussed this yet, but MPF is also a form of cyclin. So these are both cyclins, and you can see that MPF peaks first, and then cyclin follows a little bit afterwards. We have, we've genetically sequenced, obviously it's several species, but we've also looked at the amino acid proteins sequence of these cyclins and have looked at how they get degraded. And so what they think is going on here is that MPF is when something called CDK and cyclin bond together, and you get your mitosis promoting factor, and then it goes through mitosis, and as soon as mitosis is complete, then the cyclin gets degraded and you still have the CDK. And so the CDK is just by itself like a single block until the G2 checkpoint, where it binds to a cyclin and then starts the whole process over. And so they've isolated mutants in yeast and identified the genes necessary for this cycle progression of cyclins. And they've also, again, noticed some checkpoints. Again, DNA synthesis and this G2 checkpoint, where it's committed to divide. So MPF is a cyclin, and it's bonded to a CDK. Why do I say CDK? I say CDC. Wow. So CDC2 in this case, it's very specific, is a protein kinase, and when it binds to a cyclin, then you get MPF, and that's where the mitosis phase. So I think this diagram does a way better job than all the words. So cyclin, CDK, that's the CDC2. And when these two bind, then you get MPF. So as a result, MPF is a cyclin-dependent kinase because without the CDC2 and the cyclin, if they're not bonded together, you have no mitosis. And when they are bonded together, then you get prophase, which is the chromatin condensation. The nuclear envelope breaks down. Your Golgi apparatus kind of dissolves a little bit, and then you get your spindle formation. So they've actually tied the mitosis promoting factor to these events. All right, so I'm going to talk about 3 checkpoints in mitosis, and the first one is the G1S checkpoint. It's kind of the primary point where whether or not the cell wants to divide. Some external factors that can halt the G1S checkpoint, growth factors. Does it have enough nutrition, enough nutrients, enough building materials, and then is it big enough to divide? Second one is the G2M checkpoint, so right before mitosis. This is where we look at CDKs. Are they active? Once we get through this checkpoint, it's committed to mitosis. It's another checkpoint because it looks at the DNA and then asks itself, hey, did I replicate this DNA correctly? And if not, it does have the ability to go back and fix those mistakes. But then we're also going to talk about what happens when cells don't fix that mistake, because maybe they have a default gene that doesn't produce the right protein that checks the DNA correctly. So. And the third checkpoint is the spindle checkpoint, where it's like, hey, do all the chromosomes have a spindle attachment? So that way when we reach anaphase, they'll split equally. And sometimes that's not the case, and so we'll talk about, with our karyotype labs coming up here, what happens when chromosomes are not divided equally. So here, in summary, are the 3 checkpoints. The cell growth checkpoint, which is the G1S, okay? The DNA synthesis checkpoint, so after synthesis, right before G2, hey, did my DNA, replicate correctly? And then your mitosis checkpoint with the spindle fibers. All right, so what happens when the cell cycle gets out of control? Usually cancer. Cancer, by definition, is just cell division out of control. They call it cancer because, anyone here a Cancer for their zodiac sign? No?
What month is that?
July?
Yeah.
You are? Are you Cancer?
Oh yeah, what cancer?
Okay, so like what's your animal kind of symbol for the zodiac? You guys don't know? What is wrong with you? You know, like Aquarius is the water bearer, Leo is the lion. I'm a fish.
Aries is the ram.
Yep, Aries is the ram.
Cancer.
Pisces is a fish.
Cancer's
I
the
didn't know
rat.
it was a ram.
The what?
I thought it
The
was
rat.
A rat? No, it is not a rat.
Is it Cancer?
I got it up.
Herman Norcross, do you guys know the symbol for Cancer? What animal?
You're the crab.
Oh,
You're
that's
the
why you
crab.
went like that.
Yeah, you guys are the crabs. Okay, so
Oh.
what happens is that cancer cells pinch healthy cells to death because they grow out of control.
Well, that'll help me. That will help me.
Yeah, and so that's why they call it cancer. So it says unrestrained, uncontrolled growth, and it literally pinches healthy cells to death. There are some genes involved with cancer. So we have tumor suppressor genes, which means that these genes inhibit cell division, and if they're working properly, a cell will not divide uncontrollably because these genes will stop that process. So we have tumor suppressor genes that are active in us right now, hopefully. So here's a tumor suppressor gene. If it's on, if it's unmutated, it produces normal growth inhibiting proteins that keeps cell division under control. If there is a mutation where this gene is faulty and now we have a non-functioning protein, cell division is not under control, it's out of control, and we get cancer, hence tumors.
Now that mutation must be pretty common.
Well, hold on, I'm going to talk about that. So, yes, if you have a mutation there, that's one strike against you, but you have to have a second strike in order for cancer to occur. And that is the P53 gene. So the P53 gene is a tumor suppressor gene, key role in the G1 checkpoint. It checks damaged DNA, and if it detects damage, it will stop cell division, literally in its track, until it repairs that. And so if there's damage to DNA, P53 is active, it will halt the cell cycle, go back, fix it, and continue on, and then repair it. So it tries to prevent this DNA replication from taking place. However, again, if the damage is too great, like if the p53 gene is absent or damaged tumor cells, and you have that tumor suppressor gene mutation, cancer will result. That's what I'm trying to say. So you kind of have two strikes. I want to back up a little bit. So if p53 is working correctly, it will either repair the DNA. But if the DNA damage is too great, it will stop the cell cycle and tell that cell to undergo something called apoptosis, which is cell death, cell suicide. Okay? And the whole reason for that is it wants to maintain genomic integrity. It wants to make sure that its DNA is correct so that we don't get problems down the road. So we'll talk about apoptosis here now, I think. I remember right with my slides. Nope, come back to it. All right, one more gene factor that you have to have in order for kind of cancer to take over is proto-oncogene mutation. So we have proto-oncogenes in our chromosomes, hence this little blue band, you can see it. And if it becomes mutated, it's no longer called a proto-oncogene, it's now called an oncogene. And oncogenes, if they're not, if they're working properly, it will not receive the message to divide unless it should be divided. But if you have that mutation, then it's just constantly going to divide. So, to recap, you have to have a mutation in a tumor suppressor gene like p53. And then the second strike is a mutation in the proto-oncogene. So p53 is an example of a tumor suppressor gene.
Like smoking cigarettes enhances that or what?
Well, yeah, because cigarettes is a mutagen, and I'm actually going to get to that. So here's my proto-oncogene, and it's off, and I have normal cell growth division, but if it gets mutated, then it turns on, and cells just start getting crazy. They're going to be abnormal. You're going to get a bunch of tumor cells, this uncontrolled cell growth, pinching healthy cells to death. So mutations, how do these mutations occur? A lot of times, mutations are brought about by your environment. Okay, so mutations in tumor suppressor genes might allow the cell to divide uncontrollably. Mutations in the proto-oncogenes can stop working properly, and that's why proto-oncogenes become oncogenes. So what are some mutagens? Well, mutagens, any chemical in our environment that can produce a mutation, like when I start the school year, I have to watch a Rights to Know video because I am exposing myself to chemicals. So I need to be aware, hey, did you know this chemical has high risks of blah, 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 blah. Just so you know, make sure you wear your protective gloves and your goggles and blah, 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 whatever. Our custodians have to because they're dealing with chemicals every day. And who knows what those chemicals can do to you. So are you using those chemicals in a proper way? Carcinogens are specific mutagens that have been shown to actually cause cancer. So yes, cigarettes contain carcinogens. They can alter your DNA, disrupt proper regulation of the cell cycle. So like I said, you need more than one mutation. You have to have both in the proto-oncogene and tumor suppressor gene. And if so, yeah, then you're probably going to develop cancer down the road. And the reason for that is because if you have a mutation in your tumor suppressor gene, it's defective, so it can't suppress the tumors. And that's why cancer, one of those symptoms is or are tumors. Okay, now going back to p53, it can correct the DNA, it can halt the cell cycle process, or it can tell the cell to commit apoptosis, which is programmed cell death. And the word apoptosis means fallen away, kind of like a scab or a leaf. It's a orderly deconstruction. It's very clean. The cell literally shrivels up and breaks off into little blebs, and then lysosomes come and destroy those blebs. It's way different from necrosis. Necrosis is when your cells become damaged and everything just swells and bursts. Like when things get swollen, it's just like the inside cell parts are out. They're just out there. It's just messy. But apoptosis kind of shrivels in on itself, breaks off into blebs, and then you have your lysosomes that come and destroy it. So here's a normal cell going through apoptosis. Starts to form tiny little blebs, breaks off in these. Oh, this is necrosis. Oh my gosh. Anyways, but yes, we have We have vesicles leave in, it becomes ruptured, it's really messy. This is the blebs, wow. So comes in on itself, there are the tiny little blebs. This is a giant phagocyte, or maybe a giant lysosome, and it's eating these blebs and recycling the cell parts. So it's very clean, versus here you have cellular material on the outside of the cell, causing a lot of inflammation and swelling. So why apoptosis? As you guys know that when you're an embryo, your cells do commit apoptosis. Like this is the reason why we don't have webbed hands or feet, but maybe some of you do.
Oh, and
And if you,
tails.
and
Wait,
if
how
you,
do you know if you have one?
you would know.
Okay.
I had a friend who had like a webbed toe. And
That's
so those cells did not go through
Was he a
apoptosis.
swimmer?
She was not.
Sam always brags about how many things still so wet.
Why do you brag about?
Okay, so human embryos have webbed fingers and toes at the early stages, but they disappear because they go through apoptosis. Vertebrate embryos produce more neurons than needed, so they kind of overcompensates. But then, as soon as your brain starts to make connections and the, kind of set a circulatory pathway, the neurons that don't get used go through apoptosis. So that's why it's really important that you keep yourself active. You do the puzzles. You challenge yourself and not just binge watch Netflix or Grey's Anatomy
Or Matt?
I don't do.
or Fortnite. Oh my gosh, yeah.
Except that actually is kind of stimulating your brain, though.
Is it?
Wow.
But I'm just saying,
Probably
you need,
more watching
you need to
shows.
diversify yourself. Maybe read a book. It has, like
I'm
a book. It's
I'm
made of
still
paper.
on
It's got
Four.
pages.
I'm still on page 12 of 1984.
I'm
the
halfway
beginning of
through
the
two
year.
different books.
Did you know that wasn't his real name?
Orwell?
That's not his
Just
real
his
name.
pen
It's
name.
not. Okay, so apoptosis is a highly conserved process. What it means is that it's very clean. We have genes that say, hey, we're committed to apoptosis, so let's start breaking things down. DNA kind of breaks down, chromatin condenses, forms tiny little crescents, get into those blebs so that everything kind of gets recycled into tiny little packets. So we have proteases being activated that break down parts of the cytoskeleton, because the cytoskeleton is mostly protein. So we get these cell fragmentation. Like I said, it's doing this without inflammation. There's no messy cell parts being released, released outside of it. Okay, I'm gonna flip on the lights.
The first song.
Okay, so what you are gonna do for the rest of the hour is you're gonna go to a website and
Ooh, I think this is a record. My iPad's not dead after a week. The 4%.
you should have extras.